So what is muscular dystrophy? It is a genetic disease that causes loss of muscle and usually occurs in males. There are several types, but we will just be looking at Duchenne. This is caused by a mutation in the DMD gene and is X-linked recessive. This means that it takes 2 copies of the affected X for girls to experience muscular dystrophy. With 1 copy of the normal X, X inactivation leading to mosaic allows enough of the protein to be produced for normal cell function. Guys are not so lucky as they have an X and a Y, which means that if they get one of the affected X, they will experience muscular dystrophy. So what is CRISPR? It is clustered, regularly interspaced palindromic repeats. It is found in bacterial DNA and is copies of viruses that the bacteria uses as a database for future reference. Cas9 is an RNA-guided DNA endonuclease associated with CRISPR and acts as molecular scissors. Scientists use RNA to find the spot in the nuclease where they want to edit, and then that RNA shows Cas9 where to cut. Cas9 snips the DNA, causing a double-stranded break. Sensing the problem, the cell jumps into overdrive to try to fix the damage, and either disables the problem gene or even inserts a new gene, but this is a little bit more complex. This can be accomplished by attaching a new genetic sequence to Cas9. Here, Cas9 unzips the double-stranded DNA, and the new RNA strand pairs with the unzipped DNA. But more about dystrophin. The DMD gene codes for dystrophin, which is used in skeletal and cardiac muscles, as well as the nerve muscles of the brain, but not much is known about the use of dystrophin in nerve cells. In skeletal and cardiac muscle, dystrophin forms a protein complex that strengthens muscles by anchoring their cytoskeleton to the extracellular matrix. So what happens if there isn't enough dystrophin?
So here we have a visual comparison of a normal dog, a dog who suffers from dystrophy, and a dog who has shown 92% improvement in the dystrophin protein. This is mainly in the heart muscle, and although we have a 92% improvement, which is pretty amazing, we have not gotten good improvement in the tongue muscle. The tongue muscle has only shown 5% improvement. However, this heart muscle improvement is probably more important than the tongue, because if we want to incorporate this type of improvement to humans, most humans who suffer from muscle dystrophy die due to heart failure.
The deficiency causes sodium channels to dysfunction, allowing too much water into the cell, which then causes cell death by explosion. These dead cells are replaced with adipose and connective tissue, which aren't much use as muscles. Now we've learned the basic mechanics, and it's time to move on to the study. This was the first time that CRISPR-Cas9 system was used in a large-scale mammal to successfully treat muscular dystrophy. 2 one-month-old beagles were injected with a gene-editing product, and 8 weeks later, their levels of dystrophin were 92% of what would be expected in a non-mutated puppy, making this the most successful study to date. Although muscular dystrophy does not have an immediate impact on agriculture, creating an effective treatment option for the disease could help the 1.3 million boys suffering worldwide lead happier, healthier lives. And this brings us to the end of our video, so thank you for watching.